So this is the last presentation
For you.
for CC, and I would like to take the opportunity to thank all the collaborators and again Asunta for inviting us and making this possible. Thanks very much, Asunta.
It's an obligation because it's the end of the project, but okay.
Okay, so these are the institutions, and I just would like to mention that it's a mix of research institutions and very important collaborators from the producer side, producer from Tabasco, and Eco Productores, which is part of the Tropicsafe project. And I would like to mention that Rosario and Marcela are very hardworking and very dedicated for the benefit of small farmers, particularly women. And so it's a pleasure to work with them. Well, background, the little yellowing disease is trying to be managed. And for that purpose, what is being done is summarized here as it is being used in Jamaica. Surveillance, elimination of symptomatic plants, replanting, weeding, control of vectors, and take care of health. But I think the most important aspect here is the use of plants that can stand the phytoplasmas and do not get the disease. Well, in order to have plants that are resistant, Jamaica started screening germplasm a few decades ago, and they found that the Malayan dwarfs were very resistant. The Panama toll were something in between, and then they started using Malayan dwarfs, and then eventually hybrid between these two. Then we followed the example of Jamaica Coconut Industry Board, and we carried out that type of research in Mexico, testing the Pacific tall ecotypes that we had in our country. The reason was historic. I mean, centuries ago, they started bringing that material to Mexico from Asia, mostly for Philippines. So we thought that it should be some great diversity, and also following the data from Jamaica that Panama toll was resistant. Well, we have a go. So here you can see that the most susceptible Atlantic toll is the one dying in the larger percentage, and then Malayan dwarf in smaller percentage, and two of the Mexican Pacific toll ecotypes were doing very well. Okay, then we also had another test in Tabasco. The previous one was in Yucatan, and this one was in Tabasco with the hybrids, which were brought from Ivory Coast and some produced in Mexico. And these are the results, and there were 4 which were very good at, or 5 of them very good at dealing with little yellowing, but also the losses due to palm weevil were not were low. So these are good materials, and these are the most important efforts in this aspect. And within the project, we started also more of this. So we are doing 2 kind of activities within the project. First, Tropicsafe trials, which one was established in Yucatan, and we continued one that had started before the project in Tabasco. And at the same time, we were monitoring different locations where materials of interest were planted in other states of Mexico. So regarding the sites with evaluations corresponding to the Tropicsafe project in Tabasco, we have the Ojochal site, in which there is a Brazilian green dwarf, Yucatan green dwarf, Malayan yellow dwarf, and a local material developed there, and no losses due to LY during several years. In the Yucatan Peninsula, in a town called Ticur, there are a site with testing called Chom Copte, and it is divided into two, 1 and 2. In the first one, we have Yucatan green dwarf, which are already producing, and the other one we have Brazilian green dwarf and Alto Saladita, which is a Pacific toll of interest in Mexico. And this is very young. The plants are now maybe 2.5 years after being planted. No losses except in the Yucatan green dwarf trial, but it was only 0.5%. I mean, it was 3 plants of the whole lot. But this is actually a good result because it is telling us that the disease is present. Then we also included recently monitoring of the site, which I mentioned already at least 2-3 times, Caridad del Cobre, where there was a population of segregants and Atlantic toll. In this one, we were expecting losses, not as high as in the case of the Atlantic toll, and that was the case. The hybrid by hybrid, it was about 50%, and the Atlantic toll was nearly 100%. Because of that, we planted the Brazilian green dwarf next to it. It's already been there for maybe 2 years, and no losses due to LY. So it seems that the materials that are placed here, except those in this case, are surviving. And the fact that even in the case of the site where these very large losses took place, we have some materials like this one that we just planted, but there are also other I think it's in the next one. No. Well, I'll go back to that later. And these are to show you the Ojo Shell trial, the plants as they are now, and how the materials are, the setup of the materials. This is to show you that where the trial is, and next to it is the site where the hybrid trial I mentioned before was, or well, it is there still. And the importance of this is that there been Freddie and colleagues have detected little yellowing in plants and insects, but also there were losses of plants. So the disease is present, and there are other phytosanitary programs as well. Then in the Chumcopte trial, this is the setup, and these are the 3 plants that were affected. These are the pictures. The plants are gone now, but it's only 3 out of nearly 600. Then this is the new one. The plants are like this now. These are pictures taken only a few weeks ago. And this is a picture of the Caridad del Cobre place where plants died in large numbers. And what it is interesting here is that this is the site. This is where Caridad del Cobre is. In red, the Atlantic toll, and you can see that within 1 kilometer, you have Yucatan green dwarf present, Pacific toll present. This is the initial test that I showed you before, and these are still there. No losses, not one single loss, even with this very strong outbreak taking place there. So this is something that we can consider as showing that these materials can stand the disease well, and that whenever you plant susceptible materials, you are going to find losses. And there's no other way it is going to happen, even if you don't see any places with important incidents or no incidents at all. So this is, we believe, due to the fact that, as mentioned before by one of the presentations I did and some of my colleagues, that you have phytoplasmas present all of the time in many plants that are there. You don't even know all of them, but that is impossible to avoid. We cannot get rid of that. So they are there forever. So what we need to do is to avoid the planting susceptible materials. Well, in regards to the monitoring of other sites, in Colima and Guerrero on the Pacific coast, we have these materials, no losses at all. In Veracruz, we have this material, no losses at all. But in the site, we had 2 cases of Adonidia merili palms dying due to little yellowing. And in Yucatan, Merida, we have Pacific tall, no losses. San Crisanto, as I just mentioned, we have Pacific tall, Yucatan green dwarf, and Brazilian green dwarf. No problem at all. Well, this is just to remind you about where these sites are. And since it seems to be a good way to go to screen for resistant material and then use it for replanting. So the next thing that we have to do to facilitate this is propagation in vitro in a fashion that is highly efficient. So we are doing that at CC, and these are plants produced in vitro, and some of them were taken to Cuba. And these pictures are actually when the plants were already in Cuba. And these are plants, some of these plants shown here in the field. This picture is nearly 1 year old, so they must be now much larger. And then, regards to conclusions, well, first of all, by testing coconut germplasm for LY resistance, part of the Tropicsafe project and other projects previously, we are learning which ones stand LY and could be used for planting purposes and management of LY. We need to continue increasing the genetic diversity of LY resistant coconut germplasm by introducing new materials. We have done that already, and testing them to detect LY resistance in the field, and hopefully very soon using more powerful tools like molecular markers, as shown before in the meeting, or as mentioned before in the meeting by Andrea Peravito. Since replanting with coconut selected for LY resistance and obviously other qualities as well, is the best way to go to manage coconut health and yields. It is very important to support propagation of selected coconuts using biotechnology for trying to meet the very large demand that it is currently existing. I'm talking about ICC, International Coconut Community, is mentioning that we need to replant at least 6 million hectares. And I'm not telling you how many plants go there, because I cannot imagine the number. It's very huge. Well, the next 2 slides are to, with the purpose of asking you to keep them in mind. One, this is to show you what happens if you plant susceptible materials. We don't want that. This is to be avoided. The following one is the contrast, and that is what happens when you plant plants that are resistant to the disease. And these plants are next to a site with high incidence in Tabasco. The plants are still there after 20 years of being planted. So that's it. Thanks very much.